The Ontario Diagnostic Days on realagriculture.com is brought to you by the Ontario Ministry of Agriculture, Food and Rural Affairs, the University of Guelph, Ontario Soil and Crop Improvement Association, Grain Farmers of Ontario, Agris Co-op, BSF Canada, Bear and DeKalb, Corteva and Pioneer, Great Lakes Grain, Mazex, The Mosaic Company, Pride Seeds, and Syngenta. And welcome everybody to Ontario Diagnostic Days, episode 8, the live version. It's great to be with you. I'm your host today, Sean Haney, and this is a special edition of The Agronomist where we kind of bring in the Ontario Diagnostic Days, and I'm really looking forward to the discussion today. We got some great topics, some very, very important topics that fit very much in line with what we tried to accomplish here in partnership with all those sponsors in terms of bringing you some of the latest and greatest in technical information as it applies to what is happening out in your field. So really, really looking forward to the discussion. Like I mentioned, we are live. So if you want to participate in today's discussion, all you have to do is enter your comment or question in the box, whether you're watching on YouTube, Twitter, or Facebook. And we really do appreciate that you are here today. We know more wet weather in Ontario, which is really creating some havoc when it comes to trying to get not only the wheat in or establish that healthy wheat crop, but also some challenges, of course, with edible beans, soybeans, and corn harvest at the same time. And then some other issues like compaction and things like that as well. So although the growing season has been quite good in Ontario, harvest is presenting itself to be somewhat of a challenge. So let's get to it. Let's bring in today's first guest. It is Dave Hooker with the University of Guelph out of Ridgetown. Hey, Dave, how are you?
Hey, I'm doing great. A bit saturated, oxygen deprived, like the wheat crop.
Yeah,
that we have in southern Ontario.
Yeah, it's a real challenge here right now.
Yeah,
But hey, there's from a positive standpoint, there appears to be some really strong yields from a corn perspective out in the field.
Yeah, absolutely. And the bean yields, I think, are way up there. Maybe not the highest yields that we've ever seen before, but we certainly can't complain about a lot of the yields that we are seeing this harvest time. So that is a good news story for sure.
As growers in Ontario are trying to get Harvest 21 completed, a portion of their mind as they're getting some of that windshield time is on the 22 crop. And one of the, I think the challenges on the 22 crop is fertilizer. Producers seeing high prices, hearing about shortages of supply. It's going to take some extra work this year to do your fertilizer planning properly, Dave.
Yeah, absolutely. And I think a lot of the inputs for 2022 will be much higher in terms of cost. And there are some inputs, though, that we cannot change very much for example, seeding rate, seed input costs. Like there's just a very limited amount of management that we can do to reduce those costs. Obviously, we can't reduce our plant population by 2. But for fertilizer, it's there's a lot of good, I think, thought processes that need to be done regarding what fertilizer to apply, when to apply it, how to place it, and the strategy for 2022 in particular. So that's a very short-term strategy. We have been talking about long-term strategies, but I think for 2022, we just have to focus on that short term.
I've had a couple growers say to me on our Real Ag radio show, when we do our Farmer Rapid Fire on Thursdays, I've had them say, you know what, I know prices are high. I got to go ahead with what I usually do. But at the same time, I have heard some growers say, I plan to cut back, and I know that potentially creates some other problems when it comes to yield loss being the first one, especially when it applies to nitrogen. From your perspective, what do growers have to keep in mind as they put together this fertilizer plan on this 22 crop?
Well, I think that we need to be aware that, like, what happens if we do cut back? So let's explore some of these questions. And I think that, we can dismiss the fact that, crops, high yields need high inputs, high nutrition. That is true. But what do the data say regarding um, if we do cut back, what will happen to our yield potential, or what is the probability of a return on investment? And I've always said, I think I say it just about every time that I come on these shows, is that farming is a probability. Like we establish management strategies based on probability of return on investment in both the short term and long term. And I just think that we should focus on that probability of return on investment, or probability to return, something based on some management decision, whether, apply a fungicide, or plant a specific hybrid, or or fertilizer use as well.
Mhm. Dave, we have a clip. You're in it, and talking about some P and K research and recommendations for 22. Let's watch the clip
Great.
because I've got some more questions, and I think our audience will as well. If you do have a question for Dave based on what you hear on this topic in the video,
Yeah.
please enter it in the comment box, no matter if you're watching on YouTube, Twitter, or Facebook. So, producer Jay, let's pull up the clip.
Of course, applying a nutrient when the response to that nutrient is low, that is not a very good management strategy. Conversely, not applying a nutrient when that crop is hungry for the nutrient is not a very good strategy either, because that could end up with a lower yield if the crop needs that particular nutrient. So we preach about soil testing a lot, how important soil testing is. And of course, with these times, with fertilizers so expensive, the importance of soil testing is higher than ever, more important than ever. Possibly variable rate application is more important than ever. So let's first discuss some strategies with phosphorus and potash. So when we look at phosphorus and potassium, and nitrogen as well, but let's just focus on phosphorus and potassium, when we want to look at the rate, for one thing, we use soil tests to predict the amount of phosphorus and potash that may be available to the crop. In the agronomy guide, and there you will find the OMAFRA recommendations for P and K. Now, these recommendations have received quite a bit of attention in the past while, especially for higher yields, because these recommendations are relatively old. So the critical values in those soil test recommendations have not changed that much. So when we look at a response curve, and when we don't get any more response to fertilizer, let's say the soil test is at some level where we don't get any more response or very little response to fertilizer, that is the critical soil test level. So let's look at some data. So I have some data from our long-term trials that really underline the importance of this critical soil test level. If we're below this level, so if we have soil test levels that are relatively low, we better apply some fertilizer, or else we're going to risk some yield reduction. So in the past few years, we've talked a lot about these long-term P&K trials that we have in Ontario. We talked about building up the soil test levels to a critical level and then maintaining those levels. So that initial build, that build process, of course, can be very expensive, and it is, of course, very expensive to do that these days. So this is not the time for a build strategy. But we also had a number of different starter fertilizer treatments in these trials. The first column shows yield responses to starter where P and K are relatively low. So we're talking about P levels, soil P levels that are about 20 parts per million or less, and K levels, soil test K levels, about 120 parts per million or less. Now, look at the bottom of the first column. We have 628.28, and this was applied essentially at removal rate. The corn with starter produced 186 bushel per acre, and the corn without starter, in relatively low P and K levels, 155 bushel per acre. So that starter effect increased the yield by 31 bushel per acre. So that is quite a significant starter effect. And so something that should be underlined in terms of if your soil test levels are relatively low, the response to a starter is relatively high. So that seems like a very good investment strategy to me. But look at that second column. Look at that second column in the data. We have soil P and K levels over 20 and 120 for P and K, and the yield response to that high starter rate was only 9 bushel per acre. So in that scenario, that 9 bushel per acre, it probably won't pay for that 628.28 that we applied basically at rates at removal level. The same story is in soybean as well. So in the low testing soils in the first column, the soybean yield response to starter was 5 bushel per acre. In moderate testing soils, the yield response was essentially zero statistically. And so in this case, in the starter, we applied the starter in a 2x2 band. But interesting enough, a lot of work that Horst Bonner, Omafra soybean specialist, has done, if there's a recommendation for P and K and soybean, a broadcast fertilizer application will do just fine. Soybeans will respond to that. But in corn, phosphorus response to a broadcast application is probably, the probability is very low in that case. So a little bit of a difference there between soybeans and corn.
Great stuff, Dave. I really appreciate you putting that video together for us. Talk a little bit more about the difference there between corn and soybeans and your approach, which you talked about right at the end of the video.
Yeah, of course, there's various strategies to apply the fertilizer. And at the beginning of the video we talked about what rate should we apply. And I'm not saying not applying anything on corn. I'm just saying that the fertilizer response or corn response to fertilizer is less, where soil test levels are high. And so that's a management strategy, something that one could consider. But placement of the fertilizer can be important as well. And we know that placement of phosphorus and potash, for that matter, in corn is more important than it is for soybean. And so that was kind of a surprise to us, actually. And a lot of the work that Horst has done shows that soybeans do respond to a broadcast application when soil tests call for P and K. So the placement factor needs to be considered, and the rate factor needs to be considered. And really, that 4R management strategy, I think it's a good way to, I guess, unravel or just make that complex situation more easily understood.
Yeah, but it all starts with the test, though, because all
That's
of
right.
all of these decisions are based on whether the test shows high amounts or low amounts of
Yeah.
residual fertilizer there for you to use. So if you don't have the test, you're really shooting in the dark, which could be
Yeah.
a very costly decision based on the fact that fertilizer prices are so much higher in 22 than prior couple of years.
That's right. And I'm just astounded that still the frequency, the amount of soil testing that people do is not very high. It's not increasing. So as the fertilizer costs increase, you would think that soil testing would increase as well, because people, it's just like, if you're not feeling very well, you put a thermometer and test and take your temperature. Well, you usually do that when you're not feeling very well. And I think we need to do the same thing on the farm. We want to, I guess, make good decisions, fertilizer wise, nutrient wise. And the first thing that we need to do, of course, is to take a soil test.
Do you have any thought or comments on the how of that? Because right now out in Western Canada, they're going through a whole different situation where, they have lots of residual fertilizer in a lot of areas because the crop, there was no crop, and so the plant didn't use
Right.
that fertilizer. Ontario is a different situation because we do have very good corn yields, in particular. Any thoughts? You mentioned variable rate application, but any thoughts on things like grid sampling versus other methods?
Yeah, there's like all of those I guess uh strategies are debatable. It depends on really how much money you want to spend, on how comfortable you are with one consultant who says like do this and another consultant says do that. And but I think that some kind of intensive soil sampling is very important. And so
Yeah.
we've been saying this for years, you should soil sample, on small land areas of the field. And so instead of sampling like a 100-acre block, you should sample, smaller blocks. And just to get an idea of the variability throughout the field. We know that soils are variable throughout the field. And I think this is a perfect time to really assess the variability in your field and to figure out maybe going forward, like you can't just throw on a variable rate application, variable rate system on a planner, or perhaps you could broadcast variable rate. But I think this is the time where we really need to consider that variability, spatial variability that we're seeing out in the field, and adjust fertilizer rates accordingly, especially for P and K.
And when you get the results, really work with whether it's your private consultant, your ag retail, whoever you're working with
Yeah.
on the farm, but uh, increase the intensity and maybe heighten the analysis of those reports in a year like this, because you got
Mhm.
like the wrong decision just can cost you so much and
Yeah,
it's not about just the
exactly. Yeah.
agronomic side of it, it's the financial side of it.
That's right. The wrong decision, if you don't apply enough, that would cost in yield, of course. And if you over apply, that will um, take an effect on the pocketbook for sure.
Yeah. Okay, Dave, you're going to come back a little bit later on in the program. So we'll talk to you here in a little bit. And thanks a lot for discussing
Right.
fertilizer
Right.
planning with us. Okay.
Right. Thanks.
Yep. Okay. Now we're going to bring in Francois Tardif. He is with the University of Guelph. Francois, how are you?
I'm good, Sean. I'm good. Trying to stay dry.
It is a problem.
Yeah.
I think a lot of farmers in Western Canada wish you could just blow it out west. They would
If we
gladly take
could, we
some of this
would.
moisture. Absolutely.
That,
Okay, we're going to talk about weed issues here. It doesn't matter what year it is, 20, 21, 22, or any year in the future. Resistant weeds are here to stay and are a problem that farmers are going to have to deal with. When you look at weeds like water hemp and glyphosate resistant Canada fleabane, palmer amaranth, um, do you have any thoughts on these three weeds and some of our challenge when it comes to resistance?
yeah. I'm, tell you the truth, I'm surprised how quickly some of them have spread. Like, water hemp we found in 2003 the first time when my cobra was called to a field near Sarnia was, group 2 resistance. And a few spots at the time, and I tried to raise awareness. And at the time, it was like curiosity. I would go around and field tours with my pot and my plants, and people would laugh, right? And not much interest. And because, well, who cares about group 2 resistance? At the time we had Roundup ready, and then we'll
Mm.
all be fine. And uh, then when that system broke about 2010, 2012, when we first start to see Roundup Ready resistance in Wareham, then it hit people hard. Now, what surprised me is how quickly it spread. And I know Peter Sigma at Ridgetown and the students have done surveys and shown how quickly that spread, much more than I thought. And that's always surprising. I mean, they ask us to predict what's going to happen in terms of which weed is going to be next, and that kind of stuff, and we almost get it wrong, right? Now, fleabane in terms of spread, that one's a bit more understandable. It's got that little seed with that little parachute. It flies around quite easily. Although it's not the only thing that matters there. I was interested in listening to Dave about like and the testing, right? Like knowing what's out there. And I think that's what we'll need to do with weeds. I'm not sure, how far the drone technology is, but going early in the spring or at different times where you could fly a field and then find out those patches that are suspicious when the soybeans or the corn is small, or at harvest when the soybean is turning yellow, and then you can see these patches instead of waiting next year when that patch got bigger, right? That's what's happening now. We get farmers calling us, and then they say, I've just noticed a weed problem. And you walk there, it's like a jungle of weeds, where, 10 acres. You go like, well, yeah, it just happened this year. Well, it can't be, right? Like there was a little nucleus, and then it started to spread. Scouting from the highway is difficult. So I'm sure the technology will help, but it's takes, a conscious effort. You need to look for these things as well.
Yeah, it's interesting you bring up new technologies because I was just kind of wondering, based at the stage that we're at here today, as some of these weeds have spread faster and further than maybe we had first thought. There was a time where Palmer amaranth people thought, would never hit a state like North Dakota, right? And now we have it in provinces like Manitoba have found it this year. So these weeds have surprised us. Um, when you look at this battle against these weeds, does it sometimes feel like we're losing the battle and we're going to need some new tools and technologies to get out in front of it again?
Yeah, I had a student come to me one year after a class, and was talking about succession and how nature responds to what we do. Like we try to do with the land something that nature doesn't want to do, and he always got quite depressed. He said, "Well, we're never going to win." So no, well, it's a constant battle because the enemy is changing, right? Like the weeds of the past, are still there, but we kind of get a handle on it. Like I was talking to Clark Branzel on Twitter, like
Mm.
exchanging on Twitter about when people were freaking out about velvet leaf in the 1980s. And now it's like something, yeah. Well, we have velvet leaf, we have tools to deal with it. Now it's Palmer Hammerhand, like the big bad weed from the south, and it's only a bad one. It's cross pollinated, it's very variable, so you get that constant genetic changes in those population, and that's what pushes that evolution and adaptation in that weed. That uh, it comes from the desert, southern U.S., northern Mexico, but it's able to rearrange its genetics so it can adapt to these conditions. And it's been introduced in Israel, different parts of the world. So I would be surprised it will be able to survive in the prairies
Mm.
if you give it a chance. So again, that early scouting, being on the lookout and doing the diagnostic test is important.
So, I mentioned water hemp and Canada fleabane and Palmer amaranth. Is there another weed that you have on your radar here? Because we have some of these weeds that we all know about and we've been talking about them. But is there another one that you're from an Ontario perspective that you're kind of looking at and being like, we got to keep our eye out on this because I'm a little bit worried about where the future leads us.
Yeah, I mean, common ragweed's always been a tough one, and on paper it should be one that would develop glyphosate resistance, and it did, but it never spread, and we don't understand why, right? It's kind of interesting when it came to group two. We group two resistant common ragweed just spread like crazy, and it led to big time problems. And then when they first found it near Windsor, it was a big deal, but then it kind of stayed there and then somehow never appeared elsewhere. So we don't understand that. For a while, I was worried about ryegrass, common annual ryegrass, which was used as a is still used as a cover crop, a bit less popular now. It's a funny plant, right? It's a turf species, it's a cover crop, it's a forage species, it's a weed altogether, and depending on which phase of the cropping system you're in, it could be different things, and it certainly is a tough one to control, especially when people try to terminate it as a cover crop. Now it's a weed that tends to be a problem mostly in places like California, Australia, southern U.S., Spain, Italy, Greece. But I start to hear more report now from places like Pennsylvania and Ohio of that weed escaping, and it
Mm.
has lots of resistance. The analogy I made in a talk once was, um, if Palmer Amaranth is like Wayne Gretzky, then ryegrass is like Muhammad Ali, right? Like the great one, but the ryegrass is still the greatest. If you look globally in terms of resistance, that's what's happening. It's a very tough one. Now, fortunately, in Ontario, I think the appetite for ryegrass as a cover crop has kind of waned a little bit because I was a bit concerned about that. There's still, like, we've been testing some fields where people have grown, have had issues controlling it, and we found it's group one, group two, and glyphosate resistant. It hasn't spread that much, but it'd be a concern. So that would be one. But I'm worried about, like, getting Palmer in Ontario and the spread of water hemp continuing.
Yeah, we got a question here from Gord, producer Jay. If you can put it up on the screen, if you do have suspicions of a problem, what Gord asks, what are the lab options for getting suspect weeds tested?
So it really depends on which weed and which herbicide. So if you have a pigweed or lamb's quarter, no, not lamb's quarters, pigweed, nightshade, foxtail that's resistant to group 2 or triazine, then there's a lab in Guelph, Harvest Genomics, that can organize that. They do a DNA test. You can get that organized with mycobra or chrysanobead, and they tend to give you a very good and quick result that could be in season. If it's a new combination, like you get a new weed, like let's say it's nightshade, and you suspect some group 14 resistant, well, we don't have the test for that yet. We don't know what's the genetic basis. So we need to do the bioassay. It's basically collecting seeds, and then we get the seed this time of the year, end of summer, in the fall. Prior to harvest is the best time before the combine gets in the field. Get those weed seeds, put them in paper bags, send them to us, or Peter Sikkema would do some testing too at Ridgetown. There might be facilities out west that do it too for Western Canada people. And then it's basically growing those seeds, those seedlings, compare them with susceptible plants that we know, and then we apply the herbicide and look at the response. So that gives us, it's a slower process, but that tells us something that a molecular test cannot tell. Molecular test is only good if we know what the gene is. So you may get a case of resistance, looks like resistant, you send it to molecular test, it comes back negative. It could be it's a new mutation, right? We don't know. So then it's a bit like COVID. Sometimes you talk about like the antibodies test and PCR and rapid tests, get these different tests, and some may be better than others, right? And sometimes a complementarity of tests is best. So we do here in Guelph the classical tests, and then it's kind of slow, but it gives you, at least a good response, good answer to what the problem is.
Yeah, and the reality is if you do have suspicions on your farm of some sort of issue, the benefits of getting that wheat, like you should do the test because long term, at least you can get out in front of the potential issue and maybe take care of it prior to becoming a lifelong problem in that field.
Oh, you wanna get away from the guessing game, right? Like it may be resistance, it may not be resistance, and some species, right? Lamb squatters, for example, we get samples sent to us, escapes, and the success rate, if you will, the positivity rate is about 50%. So a lot of cases, lamb squatters comes to us, but it's not resistance. It's something else. Could be timing, could be staging, whatever. But with pigweed or foxtail, it tends to be 90% rate, especially for group 2. And I'm sure, like, if we look at the glyphosate resistance in water hemp and in fleabane, we're in the 95% plus positivity. But it's good to know. And it's interesting, the mentality. I was talking to a colleague from Australia a few years ago. He was talking to a farmer, trying to convince him to do a test, and the guy said, "Oh, well, I'm not sure. I think I'm just going to spray simazine in that field." And Jim said, "Well, you're going to spray $10,000 worth of herbicide to test it, but you don't want to pay $100 for a test that will tell you whether
Mm.
simazine will work or not, right?" And not every farmer is like that. We get people that send the seed, they want to know, but others like, I'll take my chance.
Right.
I was good last year. I'll be good this year.
Okay, we have a clip. We should play the clip before we run out of
Yep.
time here. So, producer Jay, if you could cue clip number 2.
Good afternoon. Here we are at a farm in Middlesex County, and here you can see the density of water hemp that some Ontario farmers are dealing with. So the first glyphosate-resistant water hemp that we're aware of was from seed collected in 2014. All we knew in 2015 was that there was one farm in Ontario with glyphosate-resistant water hemp, and it was in the southwest corner of Lambton County. Glyphosate-resistant water hemp is now in 14 counties across Ontario, from Essex County in the southwest to Leeds and Grenville County in the Ottawa Valley. So this weed has spread more than 700 kilometers in 6 growing seasons. And I would have never predicted that water hemp would be found across such a large geographic area in 6 short years. Not only do we have glyphosate-resistant water hemp in Ontario, there is now 4-way resistance to the group 2, 5, 9, and 14 herbicides in 8 of those 14 counties. So that presents a huge challenge for Ontario farmers. So in terms of water hemp, the question is how did it get here and why is it an increasing problem? So if you look at the water hemp that is on Walpole Island in the southwest corner of Lambton County, it's almost genetically identical to the water hemp that's been in Ontario for more than a century. However, something happened in the last 20 years. It's mutated and it's adapted to our corn and soybean production systems. And as you can see here, it now infests commercial corn and soybean fields. Really interesting, the water hemp in Essex County is almost genetically identical to the water hemp in Missouri. So that raises a question. How did the water hemp from Missouri end up in Essex County? And in retrospect, nobody can say for sure. It possibly came in on contaminated forage seed. It could have come in on contaminated livestock feed, or it could have been moved with migratory birds. All we know is that it's almost genetically identical to the water hemp in Missouri, and it somehow ended up in Essex County. So in terms of the competitiveness, water hemp is not as competitive as glyphosate-resistant Canada fleabane, giant ragweed, or common ragweed in corn and soybean. In trials conducted in Ontario, the average yield loss due to glyphosate-resistant water hemp interference in corn is 19%. However, in our most competitive environments, the yield loss can approach 100%, such as this field here. And in terms of soybean, the average yield loss due to glyphosate-resistant water hemp interference is 43%, and once again, in our most competitive environments, it can approach 100% yield loss.
It's quite a
Nature.
beast.
It's an amazing thing, isn't it?
Yeah, yeah. I mean, water hemp is it's quite the plant. It's it's got male plants and female plants, so it's crossing all the time, right? That's part of its success because it's shuffling genes all the time. But also, you can see if seeds moving or if you changing your selection pressure, you could have, let's say, one plant that's glyphosate resistant, another one that's group 2 resistant. They'll exchange pollen, right? And then that progeny's already got the two genes, and then you get
Yeah.
one that comes with dicamba resistance from the US, and then bang, you got three-way resistance real quicker than in a self-pollinated plant.
I was just going to ask you about cross-pollinated versus self-pollinated and how
Yeah.
that impacts the spread. So I was just going to ask you about that. Do you have more you want to say about that?
Well, it's the spread will, our seeds move from place to place will be dependent on multiple factors, and then it doesn't matter whether they self-pollinated or cross-pollinated. What Peter said about forages and soil, wildlife, that can happen. Combines to me are the big culprit. Like, you can store more seeds in the belly of a combine than a duck. I'm not saying that wildlife is not, or waterfall is not moving seeds, but combines are certainly a big culprit. But self-pollinated versus cross, it's when it comes to the likelihood of multiple resistance and also the speed at which will happen. If you want to have multiple resistance on a self-pollinated plant, like let's say green foxtail or lamb squatters, you need to do sequential selection. You start, let's say, with your group 2, you apply multiple years, you get that one, then you start a selection again. With cross-pollinated, it can happen independently in different fields, and then they can interchange as you go. So it's much quicker. So that's probably why, as people found that, hey, I got group 9 resistant water hemp, well, I'll spray Pursuit or Pinnacle. That, oh, it's already resistant. Let's go to Reflex. Oh, get some population with group 14, right? And then where's it coming from? Well, it's not me, but it could be, again, that seed movement or that pollen movement that contributed to that. So in terms of water hemp and Palmer, I think that's what made them more successful and more worrisome now, because now we have that primed weed, like weed that's primed to develop resistance, that, and then we're going to throw like whatever group 27 or whatever new technology, and it's going to adapt real quick. So it forces us to be more smart, I guess, about our weed management.
Yeah, killing weeds. There was a period of time. It was a great period of time where killing weeds was easy, right?
Yeah.
You allude to it earlier. It was pretty simple, and now we're having to scrounge and scrape for any tool that is available to us. Some of those tools go back in time, and what's old is new again in our pursuit of trying to tackle some of these weeds that are robbing us of yield. Let's talk quickly here about the impact of low versus high herbicide rates and what impact that can have on this issue.
Yeah, it's a question I get, quite often. People want to cut rates, right? And then the reason is generally cost, right? You look at the label. The label says, let's say, 1 liter per acre of that herbicide, but you're thinking, yeah, it's always working good, and I'm pretty sure I could go by with half a liter, right? So that's a low rate technically, as far as the label is concerned. And if you get the same result, the same level of control, if I cut the rate and I tell the farmer you're going to cut the rate by half, but you're going to get half weed control, then you're going to say, well, I don't want that, right? I still want 100% control. So that's the goal there as far as the farmer is concerned. And when you do that, as far as the weeds are concerned, it's the same thing. Biologically, if it's half a liter or a liter, it's the same selection pressure. But if we start to cut rate and then we start to get less than optimal control, you get like your 80%, 70, 60, 50% control, then biologically it's different. And that's when the cross-pointed species will intervene. Like, if you have a water hemp plant that you hit with a rate that gives it, 50% control, that plant may be weak, but it survived. It survived because it's got minor gene for survival. A gene that may not give resistance to full rate, but at after rate, yeah, able to get by. Now you get another plant beside that survived the same dose, but for because of a different rate. Then they start to cross, and then you start to get that stacking, that build up, right? It's like tearing a sheet of paper pretty easy, but if you stack them up and you make a phone book, then it gets pretty difficult to rip apart. So the same thing, you start to get accumulation of these genes, and then you want to go back to your full rate, and suddenly it's not working because you get those multiple gene, each with small effects that cause problem. Now with a self-pointed plant like lamb's quarters, green foxtail, that's unlikely to happen because you keep it in the same plant, and if you get that minor gene, it will not exchange with another one with a minor gene. It's trying to get multiple minor genes at once, which is mathematically impossible, or less likely.
So much to think about and consider. Francois,
Yeah. Well, it took
thank
me
you so
20
much for
years
this.
to figure it out. So,
All good. I appreciate you joining us here today and
yeah,
shedding some light on this issue because it's one that continues to challenge us out in our fields every single growing season, and things
absolutely.
change and weeds adapt and we need to as well.
Yeah,
So we're going to bring you back at the end of the show. So
sure.
very much appreciate you joining us. Let's bring Dave Hooker back in from University of Guelph Ridgetown, and we're going to talk about tar spot. This is one that I don't know, going back a couple handful of years, people didn't talk about. Probably wasn't on any of our radar. We are seeing it in Ontario. Albert Tenuta has been doing a lot of work on tar spot. What are your thoughts on this issue and this disease and the potential impact it could have on the Ontario corn acre?
Yeah, well, the disease originated, of course, in the tropical climates. Corn is a tropical crop, and then it spread to the U.S., to U.S. Midwest, and from there, like I think we've learned a lot about tar spot already just by the learnings from the U.S. Midwest, and of course, we were anticipating, I guess, its arrival in Ontario, and so right now we know a little bit about the disease and what the impact could have on corn production. And in Ontario, we're experiencing that for the first hand, really, this year. And we're kind of
Yeah.
surprised at how widespread the disease has come so quickly and affecting so many fields, especially in the Southwest.
Okay, but what do you? We have a clip with Albert here, but first, Dave.
Yeah.
So when you say that, there's probably some people in the audience saying, "Well, widespread. We're potentially going to have a blowaway record corn yield. Didn't impact yield too much. What are we worried about?"
Well, you have to have some check plots in there too, right? And so, so, we're seeing like there's lots of fungicide trials, strip trials done on farms, and this year we're seeing tremendous responses due to fungicide, and some of that response is due to controlling leaf diseases, of course, and tar spot is one of them. But we're most concerned, I guess, about future years because in 2020, it wasn't we. It was hard to find fields in 2020. Actually, the first field that we found in Ontario was at on Ridgetown campus, and Albert, one of my technicians, found it, and we were just astounded how quickly this disease has affected or infected fields in 2021, and so we're just a little bit nervous, I guess, about the
Yeah.
future in 2022.
What allows it to spread so fast?
Well, part of it is residue, and the other part is, of course, wind blown. The spores are wind blown, but a portion of those spores, of course, survive in residue, and so we're thinking probably that the disease transmission or the spread of that disease is caused by both, and probably a lot of the fields last year were infected at a very low level of the disease, and some of that disease carried over from one year to the next. And I looked in our long-term rotation trials, different tillage systems, and I can't really see any crop rotation effect or any tillage or residue cover effect on tar spot in our trials. But we know from studies in the US that crop rotation plays a role, residue on the soil surface plays a role, and also the inoculum production, I guess, movement of those spores by the wind. And because it's a polycyclic disease, that disease can increase quite rapidly in a crop.
So there is the fungicide, but based on some of that research, I'm hearing from you that we don't yet know if our residue practices need to change or how we manage that residue. We're still kind of right now still asking questions.
Yeah, and I don't think we can change of course we can adopt management, we can change the amount of residue cover, but to date, I think also in the US that I don't think that is a very viable strategy to reduce the risk of tar spot infection in corn by lowering or start like plowing our farms again. That is not a useful strategy, and it doesn't result in significant reduction of tar spot infection. And so just like wheat, Fusarium head blight, when we have a lot of that disease around, we get a lot of influence from like the whole area, like an epidemic kind of concept. And I think that's what we're experiencing with tar spot, where we're experiencing those levels, and it doesn't really matter what you do in the field, neighboring fields or the general region that becomes a more important driver than what you're doing on a specific field.
Ooh, sounds like a tough one. Okay, let's watch the clip. I got more questions about this ugly beast. Jay, pull clip number 3.
Well, welcome back to Ontario Diagnostic Day. Hey, earlier this season, beginning of July, we came to this tar spot location where we've had a number of trials going on. Today, let's go back, see what happened since July till now. What's been the impact? We're going to look at management tools. What tools do we have available to us right now? We're going to look at hybrid susceptibility and tolerance. We're going to look at fungicides, discuss maybe fungicide timing as well. Hey, let's start the tour here with our susceptible check plot. You'll notice it's changed significantly since we were here last time. What we're seeing here are typical epidemic symptoms of tar spot, where we see not significant leaf area coverage. Here we're looking at 40, 50, 60% leaf area infected with those stroma, those black tar spot lesions that we see. We had seen the significant yellowing, chlorosis symptoms, as well as the necrosis and ultimately the quick shutdown of these plants. They've cannibalized themselves to the point now that you're seeing typical stalk rot issues potentially, as well as stalk breakage. And ultimately you can see that result is that, hey, the plant's not doing what it's supposed to be. The grain is not being filled, the cob is not being filled. So in our susceptible check plot, what can we expect from yield loss perspective? We know we're going to have a significant yield loss. We won't know, of course, until we harvest, but I would suspect we're probably looking at a 20, 30, maybe 40% yield loss or more because of the impact of tar spot. So we talked about one of the tools being fungicides. The most important disease management tool we have available is hybrid or variety resistance, or in this case for tar spot, we're probably talking more partial resistance. Here we've got, again, a different hybrid. This is what we would call a tolerant hybrid. No fungicide. This is our check. So this is the equivalent of our susceptible hybrid, no fungicide. You can see definitely tar spot is here. It's doing its thing, but the initial damage or the degree of tar spot is less on this partially resistant hybrid than what we saw in the susceptible. The yield loss will be less. It may be 10%, 20%, but it should be significantly less than what we saw with that susceptible. The other thing we noticed throughout this since July is that it stayed greener longer. If we can keep the plant doing its thing and staying green, filling that grain, that ultimately leads to better yields. So now let's see what happens when we put all those tools together here. Here we've got that tolerant hybrid. We've got that same fungicide that we saw earlier in the susceptible that had some efficacy, not to the same degree as what we're seeing here. You can see how combining or integrating our tools together under an integrated tar spot management can help us in protecting both the plants as well as yields. Here, first of all, they're standing much better than they were over there. We actually still have some green here, which we had no chance over there as well. Look at these ears. They're bigger. They're still upright in that. They're still filling. Anytime we can continue to fill, we're making yield, we're making money at the end. So if you look at these, again, we see a lot of tar spot in here that's come in. Again, this one application, so this again was that VTR1 application. We see that fungicides have a period of time when they're effective, and usually that's about 3 weeks or so. If we had come back in with a second application of an effective fungicide, these would have stayed greener longer. Again, that's something we've got to look at for the future. How do we build on the tools we have now, both in terms of hybrid susceptibility and tolerance, fungicide timing, the epidemiology and biology of the pathogen here, that ties into modeling, such as tar spotter that's available out there as well, that can guide us in terms of both our risk as well as our fungicide timing. But again, we have tools that can help us now. We just need to build on those tools as well.
You know, the cob comparison at the end tells the entire story. Cob size, kernel depth, kernel size, the girth of the cob, like that visual just tells you everything as far as the impact this can have.
Yes, absolutely. And so since that video was taken a couple weeks ago, and Albert has harvested several of his fungicide trials, and he has noted, like he guessed, he speculated some of the yield impacts, and he was almost spot on. Like some hybrids were 40, or some fungicide responses were 40, 50 bushel per acre, and a lot of that is due to that tar spot infection and control.
Okay, so Albert alluded to it in the video. We want to put up Gord's question here, Jay. Are there hybrids showing tar spot resistance here or in the US? Let's talk about that a little bit in terms of hybrid selection.
Yeah, exactly. So we tried to answer that. We knew that there was some hybrid response, and we can't say that, a hybrid is perfectly resistant. Like there's no hybrids of that caliber. And so we say that genetics, there's some partial resistance, at least offered by genetics. And to answer that question, Albert and I have been very busy rating the Ontario Corn Performance Trials in the past couple weeks. And so let's just see, can we just pull up slide number 2? And I've got some data analyzed, and this is the first time we're showing this, I guess. And this is from the Corn Performance Trial. And so we have a bar chart here. And the stroma are the fruiting bodies of tar spot. And so the most susceptible hybrids, like I just have them numbered here, so they're kind of anonymous at this point. But these are the results of three of our locations in southwestern Ontario, and 3 reps per location. So each bar represents 9 uh, experimental units, so to speak. And so the most severe hybrid was stroma with the fruiting bodies of tar spot. It had close to 24% or around 22% of the leaves that were covered with tar spot. And the most that a hybrid that had the most partial resistance is on the right there, and it showed about 9 or 10%. So there's some level of hybrid differences, a difference across hybrids, but of course there's no hybrid that gives total resistance. And so we are definitely, and plant breeders are working or have been working on resistance in plant breeding to at least offer some more resistance to tar spot.
And this is why the recommendation is to combine a hybrid that is effective against tar spot, but also including that fungicide in that package, that more of that toolbox approach.
Yes, exactly. So we talk about this in wheat all the time with Fusarium heblate. So genetic resistance would be the first line of resistance, and then when you combine that with like partial resistance, and then when you combine that with fungicide, of course you've got a great 1-2 punch in terms of managing Fusarium heblate. And I think tar spot just fits in with that strategy very well as well when you combine partial resistance due to hybrid, and then combine that with a fungicide when conditions are right, when the environment there's a high risk of the environment to produce tar spot. I think that's when we need to consider fungicide.
When am I going to know that you got numbers at the bottom here? When am I going to know which hybrids those are?
Yeah, so that's what we have to do. We have to, I guess, assess whether a hybrid is consistent, and so we can rate our hybrids. And so we have to. I think this information is very important for the individual seed companies, and we will pass this information on to their seed companies. We will identify their hybrids, and we will ask them, yeah, does this information fit with your rating systems and your breeding program? And then we will go that route initially, but hopefully the seed companies will have their hybrid ratings in their seed lineups as well.
What if I'm outside of the geographical zone where we are seeing tar spot currently in Ontario? Am I still, is it still my best practice to plant a hybrid that is highly effective against tar spot to try to keep the spore load as low as possible and not let it build up at all?
Yeah, exactly. I think so. I think that is a great strategy, and we know how quickly this disease has become a problem in the Southwest. And I think we just need to prepare for those things. And the best source of information for possible hybrid resistance or partial resistance is from the seed company. And so if you want to reduce that risk, talk to your seed dealer. But also, if the environment is favorable for that disease in 2022, I think we need to consider fungicide. And so there's a whole array of, I guess, decisions and strategies that you could make that need to be made when trying to make a fungicide decision regarding a fungicide application. So before we get on to another topic, I'd just like to show that slide number 1, Jay.
Yeah.
And it just shows the level of leaf disease coverage that Albert and I rated in these trials. And on the left there, there's a leaf. Of course, the leaves are all senesced, but we can still rate the hybrids in terms of maybe gives some evidence of susceptibility. And so the leaves on the left, maybe 40 to 50% coverage, and the leaves on the right, 8 to 10. And so our best hybrids that we have, at least in our corn performance trials, are the ones on the right. And so there's still tar spot there, but the best ones that we have, the leaves look like the ones on the right, and the worst ones resemble the leaves on the left. And so that's kind of the range that we're looking at.
Dave, are there ways for me to measure my spore load in my fields, or is it just a case of looking at the leaves and doing this analysis?
Yes, there's research being done by where Albert and I are involved in the crop protection network, and there's some spore models being tested, and there's an app that tests, the probability of whether you should apply a fungicide or not, and so that app assesses a number of these different variables. It's like a modeling tool that one can bring all these variables together according to our latest best knowledge of the disease, for making the most informed decisions regarding fungicide application. So there's a lot of decisions that, like, factors that go into making that decision, and one of them is just the environment in itself. And so if you could just pull up, Jay, if you could pull up slide number 3. I'd just like to show this. So here are the 3 OCC locations, and Tilbury and Ridgetown, they had an average tar spot coverage, the stroma, of just over 11%, but Dresden was in a much different environment, much more favorable environment, so it had close to double the tar spot infection at Dresden. And you can see the hybrid ranges from the best hybrid that we have to the worst by those bars at each of Tilbury, Ridgetown, and Dresden. So just by those bars, like, you can see how much of an influence that hybrid choice can have. But the environment plays a very important role in the eventual tar spot infection.
Oh, I,
Yeah.
important discussion. Thanks for sharing, Dave. I really do appreciate great work by yourself and Albert and your technicians on this one, because we're learning sort of as we go here on this, and obviously it's in the U.S. and so there's also gathering
Yeah.
some of that data and trying to learn from it, really kind of paving the road as you drive, sort of thing. So
Yeah,
really do
exactly.
appreciate your guys' work on this. Hey,
Okay.
let's bring Francois in, and we'll kind of go through our final wrap of questions. Francois, you thought you had problems with weeds. You don't. At least you don't have to deal with tar spot. You like, yeah.
Well, it's all tied together, right? It's all the same biology. It's interesting when Albert talks about putting these two tools together, the 1-2 punch. It'd be better if you had a third punch, right? A 1-2-3, right? But it's also important that these tools they be acceptable to growers, right? When we talk about integrated weed management, like we say, well, mix the herbicide with some tillage, fertilizer placement, row width, etc. Right? So some of it may be appealing, right? If I talk to farmers about mixing herbicides, they get excited, right? When I showed plots where we mix like 2, 3, 4, 5 different modes of action, say, yeah, I can do that. Like, we like to mix, but if we start to talk about increasing soybean density, maybe changing row width, well, that's not going to fit my planting pattern, or whatever reason, right? So some of it is not always appealing. In Australia, they're talking about bringing back sheep to control weeds, but some farmers say, well, I don't want to deal with sheep anymore, right? So on paper, things are pretty easy. You can have all the tools you want, but especially if you have a very useful and very efficient tool like Roundup Ready was, like, it simplified weed control so much, right? Like, one pass of Roundup and then weed control, you can check that out. Then I can deal with the other stuff, the insects, the disease. But it's always changing. And I was thinking about all this. Like, you invest so much in fertilizer, in seeds, in crop protection, and I don't know of any other business where you just throw all that and then you say to Mother Nature, okay, deal with it now. Right? You have no control whatsoever on the rain, right? Like, in some places you can irrigate, but temperature, hail, whatever, right? Like, you get too dry,
Yeah.
get too wet, what can you do? And then your whole livelihood depends on that. It's
Yeah.
quite
Yeah.
stressful, I guess.
Well, and there wasn't meant to be any sort of a theme to today's show, but as I'm having these different conversations with you guys, it's just how adaptive we have to be in response to Mother Nature's response to us, based on what's happening with diseases, and we didn't even talk about insects today, of course, but with weeds, like it's like this battle, this back and forth battle, and if you if you aren't as adaptive as Mother Nature is, you start to lose, and you're kind of losing that ground, and essentially then you're losing out in the field, and that works its way eventually back to your income statement. Maybe not at
Mhm.
the end of the day or the year, but eventually down the road. It's a really really interesting pursuit between the farmer and Mother Nature. Dave.
Yeah, exactly. And this is why I love like this industry, like what we're studying and farming in itself, agriculture, like no year is similar. There's always room for improvement. And you have to be an expert in many areas in order to do to be profitable, to be sustainable in agriculture. So it's just this challenge, I think, that drives a lot of people. Sometimes, it causes some hair loss, but 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 in most cases, and there's challenges like wet weather, Mother Nature could really throw a curveball. It could cause stress, of course, individual stress, and so we have to, of course, manage that as well, and of course, stress on families, and it all like needs to be considered together. But it's one of the reasons why I love to do what I do. It's just because it's just a challenge,
Yeah,
dynamic. Yeah.
we these challenges are it's just as challenging to the research community as it is to the farmer, because
Mhm.
people look to you guys and some of your colleagues
Mhm.
to okay, what do I do?
Mhm.
This
Yeah.
is a
Yeah.
problem. Now, what am I supposed to
Yeah.
do? And you guys got to put the data together and work
That's right.
as diligently as you can. Hey, we got a
Yeah.
question here. SSEA Healthy Soils. Jay, if you could put that question up, it's for Dave. When you hit the optimum levels of P and K in the soil, should you fertilize the rate of what's being removed from the crops to maintain that mass balance?
Yeah, so I think we just need to consider, the OMAFRA recommendations as they appear in the agronomy guide. And there we look at like it's a sufficiency approach. It's a 1 year approach into the probability of return on investment for fertilizer on corn or soybeans or wheat in that particular year. And I think that we just need to adhere to those values. And of course, those values, those recommendations have been questioned, and our data certainly supports those recommendations that they hold true today. Because a lot of that data was developed like from years and years, like sometimes 40 or 50 years old. And I just think that we need to adhere to those and just think of the probability of return on investment. And we usually, if within a 1 year, we look at the sufficiency approach. And if we're approaching those critical soil test values in terms of our soil test results, then our probability of return becomes less and less as we approach that that value. If our soil test levels are low, that means that we should probably apply some fertilizer, and some may argue, well, I'm at least going to maintain my soil test levels, and that's one strategy. Another strategy could be just to look at the sufficiency approach and look at the recommendations in agronomy guides. So there's two, I guess, strategies there. If you have the cash, certainly maintaining soil test values, I think, is important. And this is definitely not the year to build soil test values.
One of the things I had in my notes here, I want to mention Bern Tobin and all the work that Bern did throughout this year's diagnostic days. Bern
Yeah.
just putting together all the footage. It basically is his summer of putting all this together. So I really want to thank Bern Tobin from Real Agriculture for his hard work on this. One of the things that Bern had in his notes for me here, Francois, is to talk a little bit about the differences between fleabane in California versus Ontario, and how it kind of speaks to a bit of the adaptiveness and how there is some differences in can you talk about that a little bit?
Yeah, it's something that struck me a few years ago. I was a grad student with Rene Van Acker. Eric Tardif was looking at winter survival in fleabane, and he got some fleabane seeds sent to him from California, from Spain, from Israel, from Iran, and when he grew them, they looked very different. Like they were fleabane, but you could tell they were different. And then got a project with Eric Page at Canada and a few grad students involved in that, and we got some fleabane a population sent to us from California because it had a special type of glyphosate and paraquat resistance. We want to study, and we start to grow it. And I don't know if you want to show the slide. It looks very very different. Um, I think there's two slides there. It's a view from above. I don't know if it's showing up, but we could tell them apart. Um, so the one from Ontario, all the population we had from Ontario will look exactly the same. That one from California, longer leaves, narrower, like more indentations. But then when we start to grow them in the growth room at Guelph under low light quality, they don't do well. And we thought, well, they're sickly or for whatever reason. When they grow them in Harrow, in the greenhouse where it's very sunny and warm, they grow like crazy, like 12 feet tall. And the student was supposed to genetics on them, had to find a ladder to go get this, to do the cross pollination. So my point there is that although fleabane seeds can fly and found themselves in the whatever layer of the atmosphere and travel all around the world, even if you have a seed that falls from California, it will not necessarily invade, right? And I was thinking it's different from water hemp. Fleabane is self-pollinated. You tend to maintain the best genotype. And although it's a global weed, a plant coming from elsewhere may not establish itself, very well. It'd be the same as taking some wheat or corn from South Africa or the Philippines and try to grow it in the prairies or in Ontario. It's still corn, it's still wheat, but it's not the best adapted cultivar. It's probably be able to grow. You may get some yield, but it'll be pretty poor compared to others. So, but again, the water hemp and Palmer, that's different. They cross-pollinated. They doing the breeding all the time, right? They doing that exercise. So whenever they get to a new site, get that shuffling of genes, and then you get that genetic diversity that will allow them to adapt much quicker.
Very, very interesting. Fascinating. Guys, we are out of time. I really do appreciate it. Dave, all the best to you, and thank you so much for joining us here today on Ontario Diagnostic Days Agronomist Edition.
Yeah, good to be here. Thanks.
Francois, thank you so much.
It was fun. I enjoyed.
Yeah,
it was fantastic information. Yeah. Great clips, great videos, as well as the slides. Thanks to everybody that tuned in. You can, of course, go to Field Crop News and you can and realagriculture.com, and you can find some of the past episodes of Ontario Diagnostic Days from this season. So I encourage you to do that. If you are looking for your CCA CEU credits, you can definitely go to the URL that you see below, fieldcropnews.com/CCA. Once again, thanks to all of our sponsors that made this entire season possible. We really, really do appreciate making it all happen. It could not happen without you. I did mention Bernd Tobin, as well as I should mention Albert Tenuta and some of his work behind the scenes as well. You saw him today in the tar spot video. On behalf of everybody at realagriculture.com, I want to thank you for joining us for Ontario Diagnostic Days, Episode 8. We'll see you again next year. The Ontario Diagnostic Days on realagriculture.com is brought to you by the Ontario Ministry of Agriculture, Food and Rural Affairs, the University of Guelph, Ontario Soil and Crop Improvement Association, Grain Farmers of Ontario, Agris Co-op, BSF Canada, Bear and DeKalb, Corteva and Pioneer, Great Lakes Grain, Mazex, The Mosaic Company, Pride Seeds, and Syngenta.